born in 1951, there were less than 2.5 billion people in the world, and today there's over 7.5 billion. This explosive growth in the human race began a little over 200 generations ago when Noah and his family came off the ark. Has this fact of human history left its imprint on genetic variation today? The answer is yes, if the data is properly analyzed. You see, data does not speak for itself, but it must be analyzed according to assumptions. And this is where the evolutionists go wrong when they say that there's no way that current genetic variation in the world can be explained if man descended from two people 6,000 years ago. And they make the wrong assumptions, and they ignore the biblical record to reason this way. For example, Dennis Venema says genetic diversity in humans is far too large to result from one ancestral couple at any time in the last 200,000 years. He has ignored the record of Scripture and made the wrong assumptions to arrive at this. Today we're going to be looking at these topics: the 1,000 Genome database on human genetic variation, and we're going to look at a model called the created heterozygosity and natural processes model of human variation. And we're going to use that model to analyze the 1,000 genome data with respect to the HBB gene and other genes, and these are metabolic genes. In doing so, we're going to try to answer some questions. What natural processes are seen in the HBB gene? How like Noah's hemoglobin is ours? Where did God put the variants in Adam's genome? How much has the human genome aged since Noah? Were there mutations in the pre-flood world? So let's begin with the 1,000 Genome Project, which was completed in 2015. And the idea was to sequence human genomes from around the world to document human variation. When they finished, they had sequenced 5,008 genomes from 2,504 people. Everyone has 2 complete copies of the genome in their cells. So when you sequence the genomes of 2,504 people, you end up with 5,008 genomes. And free access to the database is provided. This map gives the populations that were used for the 1,000 genome database. The 26 populations are organized into 5 geographic areas: the Americas, meaning Native Americans, Africa, Europe, South Asia, and East Asia. The methods I used in this analysis were to log on to the website for the National Center for Biotechnology Information, where I downloaded data for each gene. I wrote Python scripts to count the variants and plotted and analyzed these. So let's talk about human variation in the world today. And specifically, we're going to talk about this model, the created heterozygosity and natural processes model. I get this title from a paper by Nathaniel Jeanson. The model could also be called the design diversity model, which is from a paper by John Sanford and others. The basic idea is that God put variation into Adam's genomes at about 10 to 30 million positions in his 3 billion positions of his genome. God put variation, so that most human variation today is created variation. Let's talk about it. The variants that distinguish us from one to another are largely created variants. The shape of your face, whether your hair is curly or straight, if you have a dimpled chin, floating earlobes, a widow's peak, but not only physical characteristics, but also a large number of other characteristics like abilities, like verbal ability, mathematical ability, athletic ability, fine motor coordination, and a host of other things that distinguish us one from another are largely due to created variation. If we talk about the Garden of Eden and Adam and Eve, our parents, we understand that Eve is a clone of Adam. But you can't be a perfect clone. God says that he took the rib of Adam and made the woman. And so she's a clone of Adam, but she does not have Adam's Y chromosome because she's a woman. Today we're going to be talking not about the sex chromosomes, but about the other 22 pairs of chromosomes, which we call the autosomes. Now this diagram represents the genetics of Adam in the Garden of Eden. Now the two green double helical structures represent the two copies of the genome in Adam's cells. You can think of them as two complete copies of the genome, or you can think of them as a chromosomal pair from the autosome, say chromosome 6 or chromosome 11. Now, where you see the blue bar on both copies, there's no variation. We call this condition homozygous. But where I put a red bar on one and a blue bar on the other, at that position, we call this heterozygous. And in this case, the allele frequency for the variant position is 50%. Now, what's an allele? An allele is just an alternative, one or the other. You can think of a coin as having two alleles, heads or tails. So here, the allele frequency is 50% for the variant positions in Adam. Now, let's bring Eve into the picture. Here we have Eve as a clone of Adam with respect to her autosomes. And you see that every position where Adam has a variant, Eve has exactly the same variant in the same position. And so here the allele frequency is still 50%. Now what happens if we have people descend from Adam and Eve? So let's consider a hypothetical first generation. Now this doesn't represent Cain and Abel and Seth. No, this is just hypothetical kids. And here's 4 children of Adam and Eve in this first generation. Now, in the upper left, we have this guy. He has no variant at that position because he got the blue bar from both parents. So he is homozygous at that position. He does not have the variant. And the same for his sibling on the lower right. But the other 2 carry the variant position. One got the red bar variant from Adam and the blue bar from Eve, or vice versa, and that was true of both of them. So here we see that the allele frequency has been reduced to 25% in this first generation because there's 8 copies of the genome there, 2 in each person, and there's only 2 variants out of 8, or 1 out of 4, or 25%. Now let's diagram the Garden of Eden genetics. This is a allele frequency plot for design variation in Adam and Eve. Now, we're going to look at a lot of these plots, so let me describe it. On the y-axis, we have a number of some kind. You can think of it as 0 to 100% in this case. On the x-axis, we have allele frequency, and that can go from 0 to 100 or from 0 to 1. Here I've got it as 0 to 100. And notice that I have one bar on this histogram at 50%, representing all the variant positions in Adam and Eve. The 10 million to 30 million nucleotides in their 3 billion nucleotide genome are variant, and that's represented by this bar. Now, what would happen if a population of people descend from Adam and Eve? We don't have to guess. We know what this would look like because we have computer modeling, and the best of all computer models for descent of variant positions is Mendel's accountant. Now they took Mendel's accountant and they had 10 generations descend from Adam and Eve, and then they culled the whole human race down to 3 reproducing couples. Now who would that represent in biblical history? The 3 sons of Noah and their wives. Then they had the human population expand for another 200 generations to see how these variant positions were distributed throughout the population. They also allowed mutations to accumulate. Now, this is an example of what it would look like. This is not real data, and this is not an actual run of Mendel's account. This is just my representation of what happened to those variant positions. Now, in this case, the allele frequency plot just goes from 0 to 50%. So you can see that where there once was this one bar at 50%, it's now spread through the population as people have descended from Adam and Eve and from Noah. Now the tall bar on the left represents accumulated mutations. So for today's purposes, purposes, we're going to think of the allele frequency plots as having only 3 bars or 3 bins. Each of these bars in a histogram is a bin. And we're going to only have 3. We're going to have the common variants, which are the variants that are greater than 5%. And we're going to have the rare variants, which are the variants less than 0.5%. And then there's the in-between group between 0.5% and 5%, which we're going to call the uncommon variants. Now here's the thing. The common variants, those greater than 5%, are created diversity. The rare variants, less than 0.5%, are mostly mutations. And the uncommon bin between the two is a combination of the two, or a mixture. So the common variants are mostly created diversity, and the rare variants are mostly mutations. Okay, now let's look at the HBB gene. I have drawn a picture of the number 11 chromosome, not a very good one, and I have represented the HBB gene by this green bar structure. Now, for today's purposes, we're going to think of a gene as having only 2 parts, a coding sequence and a regulatory sequence. The coding sequence is, in this case, represented by 3 exons, exon 1, 2, and 3. These exons are spliced together to form a messenger RNA, which leaves the nucleus, goes to the ribosome, and the HBB gene is translated into the beta subunit of hemoglobin. So the coding sequence is the 3 exons. The regulatory sequence is the rest of the gene, which consists of the 2 introns, the DNA between the exons, and the 5' and 3' untranslated DNA on either end. So a gene today in this discussion has 2 parts, the coding sequence and the regulatory sequence, and that makes up the whole gene. Let's talk about hemoglobin. Hemoglobin is a sophisticated nanomachine for the transport of oxygen. It consists of 4 subunits, 2 alpha subunits, which are identical, and 2 beta subunits, which are identical. And the beta subunits are coded by the HBB gene. Now, it's a sophisticated nanomachine. It transports oxygen from the lungs to the tissues and CO2 from the tissues back to the lungs. It's sensitive to oxygen tension, to pH, and to metabolic factors. So it's a very complex and highly refined machine. All right. Now this is an allele frequency plot with our 3 bins for the whole gene of the HBB gene using the 1,000 genome data. And once again, on the y-axis, there's a number. The tall bar on the left is 64. The bar on the right is 5, and the one in the middle is 4. And these are our 3 bars that we're talking about. But you know, if we're asking the question, how different is our blood from Noah's, and how different is our blood one to another today, we don't want to look at the whole gene. We only want to look at the coding sequence, because the coding sequence determines the amino acid structure of the protein. So here I have added in the coding sequence compared to the whole gene for the variant positions from the 1,000 genome data for HBB. The exons are the coding sequence, and I use those 2 terms interchangeably. The exons are represented by the red bars, and the whole gene is represented by the blue bars. Now the first thing you'll notice is the red bars are shorter than the blue bars, and that's because the coding sequence is a subset of the whole gene. There's less DNA, there's less variation. Now, when I looked at this, the red bars on the left, the rare ones are 17, 1, and 1. There was 1 common variant. When I saw that common variant in the exons of the HBB, I was disappointed. Because if there's a common variant in the coding sequence of the HBB gene, doesn't that mean that there's a common difference among us today in our hemoglobin? And doesn't that mean that we commonly are different from Noah? So I was disappointed until I found out what this common variant represented. The 1 common variant in HBB exons has an allele frequency of 28%, so you see it's a common variant. It affects the number 3 histidine position in the beta subunit, and it changes the codon from CAC to CAT. It's a single nucleotide variation. But CAT CAT codes for the same amino acid, histidine. So this is what we call a synonymous variant, which does not change the protein structure because it changes the codon to another codon for the same amino acid. We can ignore this synonymous variant and all others we find if we're asking the question about the differences in protein structure. So, ignoring that one common variant, we are left with one uncommon variant affecting 139 people. There were 5 additional synonymous variants, all rare, affecting a total of 17 people. And then there were 7 missense, 4 stop gain, and 1 frame shift variant. These are the variants that change the protein structure. The missense changed the amino acid, and the stop gain and frame shift shortened the chain. So these affected a total of 53 people. So what is this one uncommon variant that affected 139 people? It had an allele frequency of 2.7% in the whole population, so you see it's in that middle bin. But in Nigeria, it affected 14%. In Sierra Leone, the allele frequency was 12%, and in Gambia, 11%. So it's a common variant in the sub-Saharan African populations. And it wasn't found in the other populations except very rarely. So what is this uncommon variant found almost entirely and exclusively in sub-Saharan Africa that affects the beta subunit of hemoglobin? Does anyone know? Right, it's sickle cell. The sickle cell variant, it's a single nucleotide variant changing GAG to GTG, which changes glutamate to valine. It's deleterious in the homozygous state, but in a heterozygous state, it confers resistance to malaria. So here's what happened. Some dude after Noah, after the Tower of Babel, had this variant on one copy of his number 11 chromosome, and he went to Africa and had children. When he passed this variant on, they were resistant to malaria. So they were the healthiest in the village where everyone was suffering from malaria. And so that amplified this variant in the population because it conferred a benefit in the heterozygous state. This is an example of a natural process in the model. You could call it natural selection. I don't like the term, or you could call it adaptation. So now we can answer the first question. What natural processes are seen in the HBB gene? The answer, increasing frequency of a beneficial mutation, namely sickle cell. So here we are left with 209 people affected by these variants. We're going to ignore the 139 sicklers, or people with sickle cell trait, because we know what that's about, and they're largely absent from the rest of the database. There's 5 more synonymous rare variants which we're going to ignore. And that leaves us with 53 people in the database with altered hemoglobin structure. We're going to now add the alpha subunit variants. There were 10 people affected with rare variants in the 2 genes for the alpha subunit. And so we're just going to add them in. And that ends up with 63 people carried variant hemoglobin structure. So, how different is our hemoglobin from Noah's? Structural variants are found in 63 people out of 2,504, which is 0.025 or 2.5%. That means that 97.5% of us today have identical hemoglobin to the reference genome in the database. And a strong argument can be made that that reference is identical to Noah's hemoglobin. So that 95.7% of us today have identical hemoglobin to Noah. This is a very powerful argument, especially when you consider that the 2.5% of people who don't have identical hemoglobin have different variants. Rare positions are variable, so that at each position in the gene, in the coding sequence, there's better than 99.5% identity in the human population. And so we are 97.5% like Noah. Now, let's talk more about variation. God loves variation. That's why he put so much of it in the human race. He loves the flowers and the butterflies. And what a lot of fish there are. The Bible says in Genesis 1:27, God created man in his own image. Now, isn't that amazing that God, who who is a unique person, a unique being. He made us in his image and gave us each a unique palette of variants so that we all are individual. Now let's ask the question, where did God put the variants in Adam's genome? My hypothesis is that God avoided putting common variants in the protein coding sequence. And the reasoning is that metabolic proteins are high-functioning machines. Everything God made was very good. Variation in these proteins could harm their function, so God would likely avoid putting variants in the coding sequence, and he would put the design variation in the gene's regulatory sequence, but not in the coding sequence or the exons. To test this hypothesis, I looked at 37 metabolic genes that coded for metabolic proteins. We started with HBB, which codes for the beta subunit. HbA1 and A2 are for the alpha subunit. MB is for myoglobin, which is an intracellular oxygen transport protein. CS is cytochrome C, which is an electron transport protein. And the other 32 genes were coding for enzymes found in glycolysis and the Krebs cycle. So I'm calling these genes of energy metabolism. Now here is an allele frequency plot showing the whole gene variants versus the coding sequence variants. The coding sequence variants are the red bars again. In this case, I've normalized for length, dividing by the length of the exons or the whole gene to get comparable data. And on the y-axis, it's a log plot. Now we have the three bins, and you'll notice that for the rare variants, the rate at which we're seeing rare variants in the coding sequence is about the same as the whole gene. But for the common variants, there's a dramatically reduced rate of appearance of common variants in the coding sequence compared to the whole gene. Few common variants are found in the exons. In the 37 metabolic genes analyzed, comparing the rate of common variants found in the whole gene versus the coding sequence, I did a paired t-test on the results, and p was less than 0.0001, a highly significant finding. So answering the third question, where did God put the design diversity in Adam's genome in the gene's regulatory sequence for these 37 metabolic genes? Now let's talk about the rare variants found in these 37 genes. These would be the mutations that are accumulating in these genes. This is a plot, and it's a little different because on the x-axis I have allele count rather than allele frequency. And this is just the rare variants from these 37 genes. I used allele count. Allele count equals the allele frequency times 5,008. And it goes up to 25 because 25 is just less than 0.5% of 5,008. Any variants that had 26 examples in the database would be in that uncommon bin, not in the rare bin. So this is just the rare bin. Now you'll notice that it has a shape like a negative exponential. And that's because of the explosive growth in the human population since Noah. Now if we had to ask the question, which of these mutations are older and which are younger, you would say the ones on the far left are young and the ones on the far right are old. For example, that number 25 allele count bin, there were 63 variants in these 37 genes that had 25 examples in the database. That meant that these mutations were around long enough to spread to 25 people each. Now on the far left, that tall bin is the singletons. The allele count there is 1, which means there were 14,000 variant positions in these 37 genes where only one example out of 5,008 genomes carried the variant. These singletons are recent because they haven't spread to anyone. So you could write an equation for this curve, the tops of these histograms, and it would have time as a variable and be calibrated to the Tower of Babel. So accumulation of rare variants indicates aging of the genome. So germline mutations are passed to the child. A germline mutation is a mutation in the egg or the sperm so that the child has this mutation in every cell in his body. The human mutation rate is about 100 per person per generation. Now that's a round number. It's been measured everywhere from 50 to 200, and it's highly variable in one population to another. So that 100 is a round number. I looked at the 37 genes and counted the rare variants by adding the allele frequencies. I subtracted the African allele frequencies because they have a higher mutation rate than other populations, and most of the papers I read on this did not have African data. And then I extrapolated the result from the million bases of these 37 genes to the over 3 billion bases of the whole genome, and multiplied by 2 because 2 genomes per person, and you end up with an accumulation of 30,854 mutations per person today. Now, mutations are accumulating. If you expect 100 per person per generation, and if it's been 220 generations since Noah, then you would expect about 22,000 mutations in each person. The 37 genes gave us about 30,854 rare variants per person, which would make the mutation rate about 140 per person per generation if there's 220 generations since Noah. Now this number 140 is very close to 100 and well within the range of measured human mutation rates. So that means that the 1,000 genome data on human variation today is completely consistent with the human population having descended from Noah. Our genome is aging. This little girl has about 200 more mutations in every cell in her body than her grandfather had when he started life. And the accumulation of these rare variants indicates aging of the genome. So we can answer the question, how much has the human genome aged since Noah? People today have accumulated around 30,000 mutations. Were there mutations in the pre-flood world? The flood eliminated all rare variants. Think of it this way. Suppose Noah had accumulated a few mutations in his descent from Adam. These would be rare variants in the pre-flood world. But in the post-flood world, if he passed on any of these, they would be common variants. If he had one mutation he passed on to one of his 3 kids, that would make it 33% in that generation. That's a common variant. So you see, the flood eliminated all rare variants. Is the common synonymous variant we saw in the HBB exons evidence of a mutation in Noah? Well, we don't know. 46 common variants in the exons of the 37 genes. I looked at them, and the synonymous to missense ratio was 33 to 13. And that implies that these were not pre-flood mutations that became common in the post-flood world, because if that were the case, I would expect the ratio to be closer to 1-to-1 for synonymous to missense variants. And because it's not 1-to-1, this implies that God may have put design diversity into the coding sequence of these metabolic genes in the form of synonymous variants which did not change the protein structure. So here are our answers. What natural processes are seen in the HBB gene? Amplification of a beneficial mutation in the case of sickle cell. How like Noah's hemoglobin is ours? 97.5% of people today have identical hemoglobin to Noah. Where did God put the design diversity in Adam? In the regulatory sequence of metabolic genes. How much has the genome aged since Noah? We now have each about 30,000 mutations, and 2.5% of people today have altered hemoglobin structure. Were there mutations in the pre-flood world? Maybe. Conclusions are these. Common variation today is due to created diversity. The human genome is aging. Accumulated mutations indicate 200 to 230 generations since Noah. Genetically today we are 97.5% identical to Noah in our hemoglobin. The genetic data is consistent with the biblical record when analyzed by the created heterozygosity and natural processes model. This confirms what Scripture has taught us, namely that God has made of one blood all nations of men to dwell on the face of the earth.